Hi, Assalamualaikum. Good morning, everyone. My name is Nur Amani binti Muhammad Zamri, and today I would like to presenting my thesis entitled In Silico Analysis of Uncharacterized Protein Expressed in FRANCISELLA novicida. FRANCISELLA novicida is a gram-negative, facultative intracellular pathogen closely related to Francisella tularensis, a known cause by tularemia. Despite ongoing research, many proteins expressed by this organism remain uncharacterized. This study is guided by the following key research questions. The first one is, which uncharacterized protein in Francisella novicida play essential roles in its survival or virulence? The second one is, what biological process do these proteins participate in? And the last one, can these proteins serve as potential targets for therapeutic intervention, such as drugs or vaccines? The growing concern around antimicrobial resistance highlights the urgent need for the novel drug targets. However, many proteins in Francisella novicida are still uncharacterized, making it difficult to fully understand the bacterium's pathogenicity and survival mechanism. Thus, the problem this research addresses is identifying this uncharacterized protein, predicting their biological role using bioinformatic roles, and evaluating them for their therapeutic potential. This study was designed with three primary objectives. The first one is to identify the uncharacterized protein expressed in Francisella novicida. Second, to characterize their biological process, molecular functions, subcellular localization, and psychochemical properties. The third one is to evaluate their potential as drug targets, focusing particularly on their antigenicity and host non-homology. A total of 150 uncharacterized proteins were retrieved from the Francisella novicida genome using the UniProt database. The key steps in the methodology include: the first one is classification based on the gene ontology annotations. Next, identification and prediction of transmembrane proteins using tools like DMHMM. Next, comparative analysis of transmembrane protein across multiple host organisms to identify non-host homologous proteins. Next, antigenicity prediction using the vaccine to assess vaccine potential. And the last one is the protein candidates were further analyzed based on the domain structure, interaction potential, and biological pathway involvement. Among the analyzed proteins, 32.19% were involved in biosynthesis and anabolic process. The second pie chart shows that 41.4% show high activity in binding function such as ATP or lipid binding. Next, the majority, which is 58.24%, were predicted to localize to cytoplasmic components. And the last one, which is D, is 7% of the protein were identified as transmembrane protein, which are especially relevant for transport and signaling across cellular membranes. Here is a summary of the key transmembrane protein identified. Most proteins had multiple TMS domains ranging from 1 to 13, indicating strong membrane integration. They were involved in essential biological pathways like ATP synthesis and electron transports, lysosomal transport, cell wall organization, and lipid biosynthesis. Their molecular functions include ATP and lipid binding, transporter activity, enzymatic roles such as NADH dehydrogenase or KDO transferase activity. This characteristic highlights their significance in bacterial survival and metabolism, and their accessibility as membrane-associated proteins make them excellent candidates for drug targeting. Homology analysis was performed to compare the identified transmembrane proteins against host genomes. Out of 11 transmembrane proteins analyzed, 9 were found to be non-homologous to host organisms, including humans. These proteins are highlighted in pink in the original analysis. This host specificity reduced the risk of cross-reactivity and increased their safety profile as drug or vaccine targets. Using the vaccine server, the antigenicity of transmembrane proteins was predicted. A threshold of 0.4 was used to classify high antigenicity. Only 9 proteins met both criteria, which is high antigenicity and essentially for bacterial function. These proteins show strong potential to elicit immune response, making them promising candidates for vaccine development. Further string analysis was conducted to determine the potential of protein-protein interactions within the FRANCISELLA NOVICIDA proteome. One protein, which is FTSW, was found to have no significant interaction within the identified subset and may represent a functionally unique or irrelevant element. They reinforce the importance of integrating multiple in silico tools to assess the functional relevance, not just sequence or structural properties. In conclusion, 150 uncharacterized proteins were analyzed using advanced bioinformatics approaches. Transmembrane proteins were prioritized for their functional role and accessibility. Through the homolog and antigenicity filtering, 9 promising transmembrane proteins were identified. These candidates show potential as novel therapeutic or vaccine targets against FRANCISELLA novicida. This study highlights the power of in silico methods in drug discovery and pathogenicity research. I think that's all from me. Thank you for your attention. And now I would like to open to panel for any question or feedbacks. Thank you.